All right, so we're back, and I was just looking at my slides here. Okay, so we had a bunch of definitions of terms that are associated with muscle cells. Instead of a cell membrane, instead of a plasmolema, we call it a sarcolema. Instead of the cytoplasm, we call it a sarcoplasm. And then we had these myofibrils. And remember, we said that's what gave the striated look to the muscle, and we'd said that's myosin and actin. Myosin is the thick ones, and those are the ones that you see under the microscope when you look at a muscle slide. The actin are so thin you don't see them, so there's like an empty spot. It makes the muscle look striped, but it's not. It's just protein. It's just protein. Okay, so we have these, and we had mentioned that there's energy that's present, there's glycogen, and that you have oxygen there that's being held by the myoglobin, all of that good stuff. Okay, so we said that there are a gajillion in every one of these muscle cells, these, 12-inch muscle cells. In every one of them, there's a gajillion, a whole lot, I don't know how many, but there's a whole lot of sarcomeres. And a sarcomere is a Z line to Z line, and in between, we're going to have myosin and actin. And we had said those are going to interact to pull those Z lines inwards, shortening our muscle. So the myofibrils are arranged as part of the sarcomere. Okay. And like I said, we have a whole lot of these darn things. Some more of this weird terminology. We said that in a regular cell, we would talk about the endoplasmic reticulum, this system of membranous channels that are present in the cell. Well, if you look at slide number 16, look at 16, you can look at these yellow things, and you can see that those are the cisternae. They're the larger portions of the sarcoplasmic reticulum, which is the muscle cell endoplasmic reticulum. So the sarcoplasmic reticulum has these large regions that are located right at the Z lines, right? And we had said that these things, what they're gonna, their primary job is going to be to store and release calcium ions, because the magic of muscle contraction requires calcium ions. Release the hounds, release the calcium ions, and the muscle contracted. Look, it just happened. I just released calcium and my muscle contracted. And then when it relaxed, it gathered it back up. So it's this release and recovery of calcium ions into the cisternae that is responsible for muscle contraction. So on slide 16, you can see the cisternae. There are two of them right there on the Z line. And then we had said there's that pale yellow transverse tubule. And now we had said that's right between the two cisternae of the sarcoplasmic reticulum. And we said that transverse tubule, it's connected to the outside of the cell. It's the sarcolema punches in as a little tunnel in right next to the cisternae of the sarcoplasmic reticulum. And we had said at the very beginning, we said that skeletal muscle cells are voluntary. Your brain tells them when to contract. So what happens? So if my brain tells my muscle to contract, what it's doing is it's sending a nerve impulse. So we haven't talked, and the first thing we'll talk about in the next set of videos is going to have to do with the neuromuscular junction, where the nerve and the muscle meet. But before we even get there, we're going to say that there's going to be a nerve impulse that's going to be flowing along the surface of the sarcolemma, the cell membrane. And what does it do? It's bubbling along the surface, and it dives down inside of that transverse tubule. And because that transverse tubule is right there between those two cisternae of the sarcoplasmic reticulum, those sarcoplasmic reticulum cisternae, it's a mouthful, those cisternae release their calcium ions. And that's going to somehow cause the muscle to contract. So let's see where we've gotten to. All right, so I was basically talking in terms of slide number 18, where you can see the triads. 2 cisternae plus 1 transverse tubule. That's a triad, 1, 2, 3. So I was just talking about that. And then we go to, the sliding filament theory. So we had said we've got a Z line on either end, and they have actin, right? So attached to each of those Z lines, we have actin. And if you look carefully at the, let's see, picture on, okay, if you look back to slide number 16, you can see a sarcomere. And a sarcomere has a Z line on either end with actin sticking out and attached to it. And in between here, you have myosin. And so we had said there's going to be an interaction between the myosin and the actin, which is going to pull those 2 Z lines together. So let's move back ahead. And so what we call this is the sliding filament theory of muscle contraction. So when the sarcomere shortens, all those things slide together. Now, I have drawn a picture on the board because I have a whiteboard. I've drawn a picture of just one side of a sarcomere. So we have the Z line here, and attached to it we have the thin myofibrils, the actin, and in the middle we have the myosin. And you notice that we have myosin heads there. And one of the things, and I'll have to draw one more picture. I know I'm a little bit out of sequence here, but we had said that the myosin heads, there's a myosin binding site on the actins that are covered up. They're all covered up, so the myosin head can't get there. And I don't have that in my picture. If you look on slide 22, you will see that if you look carefully at the actin on slide 22, I'm going to show you. If you look carefully right there, you can see this is the actin here, and there's this long blue protein called tropomyosin, and then these little paler blue proteins called troponin. And what they do is they're covering up the myosin binding site on the actin. So that's going to be critical in contracting of our muscle. But before we get to that, I want to just sort of show you the organization. We have the myosin here with the heads, and the heads extend towards either Z line. And we have the actin, which doesn't show those 2 proteins on it. And then we had said, what keeps that myosin in the middle? Why is it sitting there in the middle? And it's because there are 2 other proteins that are holding it in place. Something called titin. It's an elastic type of protein, and it is attached to something called nebulin. And at either end, you're kind of suspending the myosin in the middle of the sarcomere. So the only two things we have left to talk about for this particular set of slides is the tropomyosin and the troponin. So when we said the magic happens when the T-tubule carries a nerve impulse into the triad. It tells the cisternate, release the calcium, and the calcium goes bubbling over to what? To troponin. So the calcium bubbles over to troponin, and what does troponin do? It binds to the calcium, that little one there and there, and it pulls the tropomyosin off of the myosin binding sites that are found on the actin. That's going to be the key to muscle contraction. So let me see if there's anything else I need to mention here. No, we've reached the end of what we need to talk about for now. And next time we're going to carry on. That reminded me of a song. Carry on, my wayward son. Anyway, we're going to be carrying on, and we're going to talk about something called the power stroke. That's where that myosin head reaches, grabs, and pulls, and then reaches, grabs, and pulls again. As you pull those two sarcomeres inward, Z lines inward, shortening that sarcomere. We're going to talk about that, and then we're going to sort of step back and talk about muscle function from a bigger view. We're getting away from the cell and into sort of the big view of muscle contraction. Yeah. Okay. Have a good one. I'll see some of you, later this week, and a few of you next week. Okay, goodbye.